students, welcome to Biomix Sub. In this today's video, we are going to discuss about the anterior and posterior axis development in Drosophila. So before going detail about the axis formation, we have to understand why this Drosophila is used as a model organism. Okay, and also we have to discuss how the life cycle is taking place in case of Drosophila. So this Drosophila is used as a model organism because it is having very much of short lifespan. Okay, so for the generation of a new Drosophila, what is required? It requires fertilization. It requires two parents, like a male parent and a female parent, and after the fertilization it will form the next embryo. Okay, next Drosophila will be formed. So here there is a female Drosophila and a male Drosophila which is present. From the female comes the egg and from the male comes the sperm, and after fertilization it produces is the embryo. After fertilization, embryogenesis begins. Okay, with division of nuclei. There is division of nucleus. After the fertilization, there is division of nucleus, and due to the division of the nucleus there will be cleavage. Okay, followed by cell and tissue movement of gastrulation and organ formation. After this the organ will be formed. Okay, next it will be hatched. This embryo will be hatched. Okay, after 24 hours after hatching of this embryo, and after 24 hours there will be first instar larva, and after next 24 hours there will be second instar larva. After next 24 hours there will be third instar larva. These are the three phases of the formation of larva. Next, after 2 to 3 days there will be pre-pupa formation. Next comes the pupa. This is whole life cycle of Drosophila. Now let us understand what are the sequential events for the formation of this Drosophila. So at first what happens, there is fertilization. First part is there is fertilization. Next is embryogenesis. So what do you mean by embryogenesis? That means division of nuclei. The third is the gastrulation. The fourth is the organ formation. Okay, so for this step what happens, that for the fertilization what is required? It requires a egg plus sperm. Okay, egg and sperm forms the zygote. This zygote, what it forms after some division, what it forms? It forms the larva. From this larva forms the adult Drosophila. In the adult Drosophila, it is divided into three parts. This adult Drosophila is divided into three segments. First is the head region. Second one is the thoracic region, and third is the abdomen region. We can see here, this is the head region, this is the thorax region, and this is the telson region, and this one is the abdomen region. Okay, so what comes in the head? What are the things it is present? In the head comes the mouth. Eye and antenna. Okay, in the thorax what is present? In thorax there are T1 segment, T2 segment and T3 segment. There are three segments in case of thorax. T1 segment is known as prothorax, T2 segment is known as mesothorax, T3 segment is known as metathorax. What happens from this T1 segment? There is only one pair of leg. One pair of leg is present produced from the T1 segment. Next is the T2 segment. So we can see here that from the T2 segment, here one pair of leg is present and one pair of wing is present. So here we can write from the T2 segment there is one pair of leg and one pair of wing. Okay, next comes the T3 segment. In case of T3 segment also, there is, we can see here that from here there is one pair of leg and one pair of halter is present. So why this halter is required? This halter is required for the balancing. Okay, next here the abdomen segment is present, and at the last there is telson segment which is present. Okay, these are some of the basic points of the Drosophila. Okay, basic points about the body structure of this Drosophila. Now next comes, what are the type of cleavage it is taking place, what are the type of egg type as well as fertilization type. So the fertilization type which is present here is the internal fertilization. That means the egg and sperm which produce a zygote, it is inside the body of the Drosophila. Next is the egg type. It is centrolithal egg. That means the yolk which is present, it is present in the center. The yolk which is present in the egg, it is in the center. Next is cleavage. Cleavage is superficial and meroblastic. So what do you mean by superficial cleavage? Superficial cleavage means the nucleus is continuously dividing. Okay, the nucleus is continuously dividing without the formation of cytoplasm. Next, division. Only one nucleus divide without cytoplasm. Division is only nucleus divide without cytoplasm division. So these are some of the basic terms. Okay, now let us see how the cleavage of the Drosophila is taking place. Now here we can see the egg. This is a newly laid egg, and in this region you can see one portion. This portion is known as the micropyle, and this is known as the micropyle end, and this is a position of the egg where it will allow the sperm to enter. Okay, it is a position where the sperm can enter, and after the sperm has been entered, it will form a zygote. It will form the zygote, and after the formation of zygote, what happens? That is cleavage. Okay, the zygote will be divided into many small cells, which is known as cleavage. So here we can see that cluster of nuclei are formed in this region. Okay, so what happens in the next stage? This is a stage 2 where the division is taking place. After certain division, until the 8th division, here the nucleus will be divided continuously, and this takes place only from the Z1 and S and M phase in the mitotic phase. But here what happens? Here there is no formation of any cytoplasm. Cytoplasmic division is not taking place in this case. Here the cell cycle is biphasic. Okay, so what happens in this case? In the next stage, in the 9th division, what happens? In the 9th division, 5 cells migrate towards this portion. Okay, which is known as posterior portion. Here we can see that there are 5 poles which are present. This five poles are the nucleus which will travel towards this posterior portion. These are known as the pole cells, from where the germ layers can be formed. Okay, from here the egg or the sperm can be produced. Okay, so here the other cells which are present, they will migrate towards the periphery. Till now there is no cytoplasmic division. Okay, next, after the 10th division, in the 10th division, what happens? They are totally present. This nucleus which were present, they were totally present in the periphery. This stage is known as syncytial blastoderm. Okay, next, after certain times, like after the 13th division, near the 13th division, here there will be each nucleus which was present over here, which has been migrated towards this periphery. Now they are forming, here the cytoplasmic division is taking place, and here only one cell, there is a jacket like formation which is formed, and in one nucleus there is a covering which is known as cellular blastoderm. Okay, this is how the cleavage of the Drosophila is taking place. Okay, so what next? In the next we will see how the segmentation of anterior and posterior axis is taking place. So what happens? Before the fertilization, there are some molecular changes taking place in the oocyte. Okay, in the egg there are certain changes which are being formed. So let us see this. So this anterior and posterior axis formation which is present, it is decided before the fertilization. Okay, so here we can see each oocyte. This oocyte which is present, it is derived from a single female germ cell, the oogonium. It is being derived from this oocyte which is present. It is being derived from a single oogonium cell. So the pole cells which were present, they were responsible for the formation of germ cells. So from a single germ cell, there is formation of oogonium. From the single oogonium, there is formation of this oocyte. Okay, before oogenesis begins, the oogonium divides 4 times. The oogonium which is present, it divides 4 times with incomplete cytokinesis. Here the cytokinesis is not taking place, only the karyokinesis is taking place. So after 4 times of division, it will form 16 germline cells, okay, which allow the surrounding epithelium layer of somatic follicle cell constituting the egg chamber in which the oocyte will develop. Here what happens in this follicle shell, this is the follicle cell where the oocyte will be developed, okay, and this portion which is present, it is near the posterior end, and the opposite end is the anterior end. So these cells which are present, they are the uncommitted polar follicle shell. They are the uncommitted polar follicle shell, and this is the posterior follicle shell, okay. After certain division, here the anterior follicle shells are present, and here the posterior follicle shells are present. So you can see here that the pole shells which are present, now they are being regulated by one protein kinases A, okay. Now let us see what happens. Here there are certain, for maintaining the anterior and posterior axis formation, there are certain mRNAs which are responsible, like bicoid and nanos. Bicoid which is present, they are present in the anterior segment, okay. The bicoid which are present, they are present in the anterior segment, and nanos is present in the posterior segment, and they are in the form of mRNA before the fertilization. They are not producing the protein, or translation is not taking place before fertilization. So these are present in the mRNA form, and they are being regulated by the microtubules. The position is regulated by the microtubules, okay. Like we can see here that here we can see the bicoid mRNA, here they are associated with the microtubules, okay. So this microtubules will allow in placing this bicoid mRNA in the posterior region. This is the posterior region of the oocyte where the bicoid mRNA is present, and the nanos mRNA in association with Oscar protein, it will be present in the anterior region, okay. So these things are being regulated by the cytoskeleton, okay. So what happens here, there are cytoplasmic polarity, that means maternal effect. In this case, the anterior and posterior axis formation is being decided, and some of the maternal effect gene, what does it? It helps in the secretion of gap genes, which are the zygotic gene. These zygotic genes which are present, they are being synthesized by the maternal genes, okay. Next zygotic genes, first zygotic gene is the gap gene, which is responsible for creating some of the gaps. After this, gap genes which are present, they help in formation of pair rule gene. These are the pair rule genes which help in deciding the pair. This pair rule genes, what is this? It helps in formation of segment polarity genes. So what is the work of the segment polarity genes? To each segment, it gives polarity. To each segment, there are small segments which are present. Here there are 14 segments. Again, it has been divided into para segment to give the polarity to this each segments. The segment polarity genes are responsible. That means to each segment, they will give the anterior or posterior polarity, okay. Altogether, this gap gene, pair rule gene, and segment polarity gene help in the formation of homeotic gene. So here in this video, we are going to discuss about the anterior and posterior axis development. This formation patterning of this Drosophila. That means here the anterior axis and posterior axis is being formed. So here we will see how the bicoid and nanos mRNA are deciding the anterior and posterior axis formation. So in the anterior portion, what was present? Anterior portion, the head was present. Next is the prothorax, mesothorax, and metathorax. Next comes the abdomen. Next, in the last is the talson, okay. So here we can see, with the help of this diagram, there is a micropylar end, and in this micropylar, near this micropylar end, bicoid mRNA is present in the cytoplasm, and this is before the fertilization, okay. And in the posterior region, there is nanos mRNA present in the cytoplasm. So after the fertilization, there are nuclear division which is formed to form the syncytium. Okay, so gradient of bicoid protein, we can see here the gradient of bicoid protein, how it is being regulated. So here in the in the anterior portion, we can see that the bicoid mRNA is by a gradient of bicoid protein is present. So here after the fertilization, the mRNA which is present, it will be translated for giving the protein. So here the protein which is formed, it is in this gradient, like we can see it is more in this portion, in this anterior portion, and less towards the posterior portion. And the nanos which are present, the gradient of nanos protein is more here in the posterior portion and less towards the anterior portion. Okay, so here after the cellularization, what happens, like acron which will be formed in the posterior portion, it will be formed in the anterior portion. Here the acron or head cap is formed. First is acron, next is head, next is thorax, next is abdomen, telson, here is a pole shell. So if we discuss about the cellularization or the formation of the body structure of the Drosophila, first comes the acron, head, telson, abdomen, acron, head, thorax, abdomen, telson. Okay, now next we will see how the before fertilization and after fertilization, how this bicoid mRNA and nanos mRNA are being regulated. Like we can see here that the oocyte mRNAs, in the oocyte the mRNA is present. So near the anterior portion, the bicoid mRNA are present, and with time it will be reduced. It is high in the anterior region, and in the posterior region the nanos mRNA is high, and gradually it is decreasing when it is coming towards the anterior region. With this, there are other two mRNAs also present in the oocyte, which are known as hunchback and caudal. Before fertilization, these are present throughout the oocyte. Okay, so here we can see after fertilization what happens. After fertilization, this bicoid mRNA which are produced, they will be producing the protein. So here the concentration of protein will be gradually it will be decreased when it reaches to the posterior pore. This is about the bicoid. Okay, next is the nanos protein. The nanos protein which is present, it was the mRNA which was present, it was present in the posterior region. Here the protein will be high in the posterior region, and gradually it will be low when it comes near the anterior region. And here there will be a interaction of the bicoid and nanos protein. Okay, there are other two proteins present over here, which is known as hunchback and caudal. So this hunchback which is present, it is present in the anterior region. The concentration of hunchback is more in the anterior region. But before fertilization, they were evenly distributed throughout the embryo. Caudal as well as protein, they were evenly distributed throughout the embryo. Okay, but after fertilization, the bicoid is having the ability that it will inhibit the caudal protein in the anterior region, and in the posterior region this nanos will inhibit the hunchback. So here we can see that there is less amount or no amount of hunchback present near the posterior region, and the caudal protein is present in this posterior portion, and it is being inhibited by the bicoid protein when it is reaching the anterior portion. Okay, so we will see how it is being regulated. So what happens in the anterior region, that is bicoid mRNA. For the formation of bicoid protein, there are certain proteins like cortex, Graujon and Stauffen, which is responsible for the formation of protein, which is inhibiting the caudal mRNA to form the caudal protein. Okay, this is in case of anterior region. We will discuss the regulatory mechanism of this, how it is being controlled. And in the posterior region, the nanos mRNA which is present, it is responsible for formation of nanos protein by the help of one protein which is known as Oscar. And Pumilio and P55, what is this? It inhibit hunchback mRNA for the formation of protein in the posterior region. Why? Because the concentration of hunchback should be more in the anterior region but less in the posterior region. So someone has to inhibit it. So the nanos protein is inhibiting the formation of hunchback protein with the help of Pumilio and P55. And in the anterior region, bicoid is inhibiting the formation of caudal protein. Okay, so at first we will see how the system is taking place. So what happens in the posterior region? So let us first start with the bicoid mRNA. What happens the bicoid mRNA which is present, it comes with another protein which is known as swallow protein and experientia protein. What this protein, this bicoid is now in the form of mRNA. If it is in the form of mRNA, it is having a 5' UTR region, and also it is having 3' UTR region. So in this 3' UTR region, what happens, the swallow protein and the experientia protein is binding in this region. So now it is forming a complex. And it is being transferred to anterior location. It is being transferred to anterior location. This is how the bicoid mRNA is being regulated to be present in the anterior region. Okay, so what next? Now next thing is that how this nanos mRNA will regulate. This nanos mRNA which is present, it is having a stophan protein and Oscar mRNA is present with it. Okay, so this nanos mRNA which is present, it is having a 3' UTR region in the oocyte. In the oocyte, it is having a 3' UTR region. Some other proteins like SMAG protein and CUP protein binds in the 3' region. It binds in the 3' region. So what is this? It will not, this SMAG protein and the CUP protein which are present, it is working as a translation inhibitor. So due to which it cannot form the posterior part. So here there will be no posterior part will be present. Okay, next what happens, the stophan protein which is present, it helps the Oscar mRNA to form Oscar protein. Stophan protein which was present, it will help the Oscar mRNA to form Oscar protein. Okay, so what is the work of this Oscar protein? The Oscar protein, what it will do, it will come and bind in this region in the CUP portion, and it will remove the CUP protein, and eventually the SMAG protein will also be removed. Okay, due to which the nanos mRNA will be converted into nanos protein. In this way, converted to the posterior portion. Okay, now the next thing is how this caudal protein and hunchback protein is being regulated. Okay, so the caudal protein which is present, so here we will see the caudal protein which is present, what happens in the oocyte. So let us now understand how the caudal and hunchback protein are being regulated by bicoid and nanos. So the caudal protein which is present, it will be synthesized in the posterior region, and the hunchback which is present, it will be synthesized in the anterior region. So these two are being controlled by the bicoid and nanos. So what happens, we will see about the caudal protein. What happens is caudal, when it is present in the first, it is present in the mRNA. So what happens, this caudal protein will be need to be produced in the posterior region, but it will be inhibited in the anterior region. So who is inhibiting it? It is being inhibited by bicoid protein. So here in the 3' region, here if it is a mRNA, it is having a 5' UTR region and a 3' UTR region. So the bicoid protein comes and bind in the 3' UTR region, due to which here the ribosome cannot bind. So here there will be no protein formation. Here translation will not take place and protein formation will not take place. So here the caudal protein is not formed in the anterior region, but it needs to be formed in the posterior region. So here in the posterior region, the bicoid protein cannot bind here, because in the posterior region there is less amount of bicoid, or bicoid is not present. Here the nanos is present. So here in the 3' UTR region, no one will come and bind. So here the translation is taking place and there is formation of caudal protein. Okay, the next thing is about the hunchback. This is the case of hunchback. So this hunchback which is present, it needs to be synthesized in the anterior region, but in the posterior region it needs to be inhibited. So here there is mRNA of hunchback. So here there is no inhibition. Here the nanos who will inhibit the hunchback. Hunchback is being inhibited by nanos, but in this posterior in the anterior region there is no nanos, so here there will be no regulation. So this mRNA which is present, it will be converted into protein, and here the hunchback will be formed. But in the posterior region it needs to be inhibited. So here the nanos protein will come and bind here in the 3' region. After that it will recruit some proteins like pumilio and braid. It will recruit pumilio and braid, which further recruits D4EH5. Okay, due to which the ribosome cannot bind in the 3' region of the hunchback, and here no protein is being formed of hunchback. In this way the hunchback and caudal is being regulated. So what happens in this case? We have discussed how the fertilization is taking place and what are the structures, what are the cleavages, what are the types of eggs, and how the bicoid and nanos are being controlled for the formation of anterior and posterior axis. Let us understand about the third metallurgical, that is the torso gene. It encodes a receptor tyrosine kinase. Torso-like gene which is present, it is encoding a MAP kinase cascade, which is receptor tyrosine kinase, and it gives a signal to anterior as well as posterior region. Like, and in the anterior region it is giving signal to the bicoid. Along with the bicoid, it is helping in the formation of head region, and in the telson, in the posterior region, it is binding with the nanos for giving the telson protein. If this torso protein is mutated, the anterior portion as well as the posterior portion, or we can tell that the head and the tail portion is being not formed. Okay, so let us see what happens if there is bicoid mutated. Here we can see here the wild type is secreting this much of bicoid, but if there is mutation in the bicoid gene, here there will be no formation of this bicoid protein, or the bicoid protein will be parallelly, it will be negligible in this case. So let us understand the mutation case. Here in the normal development, in the wild type embryo, here what happens, the bicoid mRNA is present in the anterior region and nanos is present in the posterior region. So here the wild type phenotype will have acron, head, thorax, abdomen and telson. This is the structure. Now here we will see what happens, development of bicoid deficient mutant. What happens if in this condition, this is not a wild type, here the bicoid is not present. If bicoid is not present, this bicoid was responsible for formation of acron head, and some amount was also responsible for the formation of thorax. So in this case, if there is no bicoid, these three parts will not be formed. Okay, if here there is no bicoid, that means it is here the nanos is being present. So this nanos will help in giving the tail. So the phenotype will be telson, abdomen and telson if the bicoid is being mutated. Okay, now the next condition, if bicoid is added to anterior part of the mutant bicoid. Okay, in the wild type condition, what happens, there was formation of acron, head, thorax, abdomen and telson. But in case of a mutant or bicoid, there was certain defect like telson, abdomen, telson was formed. Now what if we will add the bicoid in the anterior region? Now it will form the normal development. Here also it will form acron, head, telson, abdomen and thorax, abdomen and telson. Okay, now what happens if the bicoid protein is being present? If the bicoid protein which is present, it will be provided in the middle region. So here we can see that here the telson will be present. Here the telson was producing. Next will here the bicoid protein is being given in this region. Here there will be formation of thorax, head, thorax. Next will be abdomen. Next will be telson. Okay. Now this condition. Add to posterior pole of the wild type embryo. If this is a wild type embryo and in the posterior portion, again we are giving the Bicoid. So bicoid was responsible for formation of acron, head and thorax. Now in the posterior portion also there will be acron, head and thorax. Okay, this is how the mutation case is of the bicoid protein is taking place. Now let us see some of the genes of anterior and posterior region. From the anterior portion is for the anterior portion there is bicoid, experientia. So from this portion also there are questions being asked like what are the anterior proteins, what are the anterior posterior protein and what are the terminal groups. So here the bicoid, experientia and swallow protein are the anterior proteins. And the posterior proteins are nanos, Tudor, Oscar, Vasa, Valois, Pumilio and caudal, and the terminal group are the torso-like protein. Okay, there are some of the functions, mutant phenotype and functions also which are present. You can learn from, you can take a screenshot and see what are the factors they are responsible, what are the mutation case and what are the proposed function. This is all about this today's video guys. In the next video we will discuss about the dorsal ventral axis formation. Thank you everyone.